Hello, everyone. We recently did an experiment to see if we could reverse the effects of aging in a way. And we're going to talk about that more in a little bit. But to start out, we wanted to look at these pictures with you and just talk about them a little bit. If we look at the picture on the right of the screen, we see that this is an older gentleman. He has wrinkly skin. He doesn't have his teeth. It's a black and white picture, but we can tell that he has gray or white hair, which those indicate aging. All of us know that. We've all seen that in our lives. As our parents and grandparents get older, they accumulate these characteristics. As we look at the picture to the left, we see that these younger adults look it. They have colored hair. Their style shows that they're younger. They have their teeth. Their skin is tight. There are many different things that show us that they are younger. They're a younger generation. Now, just because they wear certain things that this man doesn't make them biologically old. I think we all know that's not what makes us old or young, the things that we wear. Now, there are biological things that do make us old or young. And we want to talk about a few of those. So what makes us old? We're not 100% sure. We all know when someone's old just by looking at them, but why are they old? There's a few things that can determine or make us age. One of those being skin problems. As we live throughout life, the sun, the UV light can degrade the skin and cause it to be spotty, ruins the elastin in the skin, so it's not as tight. Those are things that can cause us to age and look like we are aged. Another one that might not be as known is the ends of chromosomes called telomeres. As a person ages, they will start to disappear through wear and tear, and the body eating away at that telomere will start to eat into the actual DNA. The telomere is just there to protect the chromosome. And as we age, that telomere will disappear, which will then eat into the building blocks of our DNA, which means we won't have the things that we need to rebuild the body to where it was when we were younger. That is also a sign of aging and can make us age. Another is red blood cells. All cells in the body, they die. They get old, they die, and over time, it just puts a wear and tear on the body, causes the body to age. We want to focus on kind of the heart and blood and the arteries and go in a little deeper into that and how that causes us to age. One of the reasons that we age, on top of those other three, is arterial inflammation, which is what we did our experiment on. This is what we wanted to test to see how it makes us age and why it makes us age, and if it's in some ways reversible, if we can reverse this cause of aging. Arterial inflammation is the leading cause to cardiovascular disease. It's a major disease in the world today. If we look at the graph on the right side of the screen, we can see that 32.3% of the deaths in the world are due to cardiovascular disease. This is a very large amount. And this happens, a major cause of this is because of arterial inflammation. As we see in the left picture, there's a plaque buildup inside of these arteries. And as that plaque builds up, the blood cannot travel through the arteries as well, making it harder for the heart to pump blood to the body. And if the heart is pumping harder, faster, constricting more, throughout life, then it's going to wear out faster, causing us to age. So we wanted to experiment with this and see how can we decrease the arterial inflammation in our bodies to keep us more healthy and not age as fast. So is aging reversible? Well, that's what we did our experiment on, to kind of figure that out, to see, is this reversible? Is there ways to make us younger as we get older in number? We found that there is a way to reduce those side effects of aging.
So what we looked at with the inflammation is kind of this inflammatory cascade. And so in the large arteries and atrial tissue, there's different proteins that cause this inflammation to happen. And the first one being nuclear factor kappa beta kinase, or abbreviated IKK. And when this is phosphorylated, it phosphorylates, it's a kinase, so it phosphorylates nuclear factor kappa beta, which then allows more cytokines, these inflammatory cytokines, to be present. And then more of these cytokines present cause more nuclear factor kappa beta to be phosphorylated. And so these cytokines that we looked at in the atrial tissue and in the aorta and the large arteries were interleukin 1, interleukin 6, interferon gamma, and tumor necrosis factor alpha. And this was important because these are the cytokines that cause an immune response, or rather they recruit those macrophages and T lymphocytes to allow an immune response to happen. And so for us to look at those macrophages and T lymphocytes, we have to describe a concept called infiltration. And infiltration is just the presence of these macrophages and T lymphocytes because of some foreign substance. So with inflammation, there's dead cells, and these dead cells release toxins, or they're damaged, so they release toxins, or they have other things that are causing these pro-inflammatory proteins that I described previously, the cytokines, the nuclear factors, to be released. And then because they're released, the cytokines then recruit the macrophages and T lymphocytes. So this first picture shows no infiltration. There's no green spots really to show that there's infiltration of macrophages and T lymphocytes. This next picture shows that there is a lot of macrophages and T lymphocytes due to the green color present in this cell. And that shows that there's been an inflammatory response. So our study, we wanted to see the effects of aerobic exercise on arterial inflammation, mainly due to the large arteries, the atrial tissue, and the aorta. So we got groups of mice and we put them in 4 different groups. We had young and old control groups, and they didn't have hamster wheels. And then we had a young and old group that had hamster wheels, and we called that group the voluntary running group. And basically we tracked how long the voluntary running groups ran each day through our data on a chart. And then after 14 weeks of watching the mice and collecting data, we euthanized the mice and then cut the aortas, the large arteries, and parts of the atrial wall, froze them in liquid nitrogen, and then after a couple of days, we went and used some cell staining techniques. And these are our pictures showing the difference of the cell staining. So on the top, you have the control group of the old mice, or in other words, the mice that didn't run. And you can see that there is a bigger presence of infiltration happening, or in other words, the brown spots that you see on these two pictures in A and B are the presence of the macrophages in the T lymphocytes, meaning that there's more inflammation when these mice didn't run. If you look at pictures C and D, these pictures don't have many brown spots on them. In fact, they look mostly purple. So it seems like there's something going on with the inflammation, and that there's less inflammation if these guys run. And so just using basic cell staining techniques, that's how we got our data. And Jeremy here will explain the results of that data.
So, as Raf introduced earlier, in that inflammation cascade, we have this protein, IKK, that will start the inflammation cascade. As we can see in this graph, there are 2 bars on each of our variables. There's 2 bars for the young running, and the old running. One of the darker bars, the phosphorylated IKK, which means that those phosphorylated proteins will be able to activate the recruiting protein. And if we look at this data, we can see that the young and the voluntary running are about equivalent. There isn't much difference between the unphosphorylated IKK and the phosphorylated IKK. But there's a quite stunning difference between the old and the old voluntary running. The amount of phosphorylated IKK drops significantly once you introduce aerobic exercise. So with the drop of phosphorylated IKK, there will be less recruitment of the cytokines. And talking about these cytokines, we can go through and look at these cytokines that Raphael introduced earlier. If we look at interleukin-1, we see on our y-axis that interleukin-1 was measured in picograms per milliliter. And a picogram is 1/trillionth of a gram. So it's pretty small units, but that's what you need to measure proteins. So we see that same pattern though. The young and the voluntary running young are about the same. There is very little difference, but between the old and the old voluntary running, there's a dramatic drop in cytokine activity. And as we look through the other 3 cytokines that we introduced, the cytokines that we stained for, we'll see a similar pattern where the young and the young voluntary running has very similar amounts, and the old and the voluntary running old have a significant drop in pro-inflammatory cytokine activity. And here's the last one. You can see that same pattern. And these cytokines, they act as signal molecules to recruit the macrophages. Just as Raphael introduced earlier, this is what we use that staining to locate these macrophages. Macrophages are immune cells that cause further inflammation. And these cells are what prolong that inflammation. As we can see on the y-axis, we're reporting the cells and the percent of the total cells that are infiltrated. But we see a similar pattern between the young and the young voluntary running, them being very similar, and the old voluntary running, with there being a dramatic difference, a dramatic decrease once we introduce this aerobic exercise. And all this data circles back to what was introduced at the beginning. What can we do to help reverse that aging process? With what we've seen today, we can kind of start to see the effects that aerobic exercise has on inflammation and how we can reverse those effects if we introduce aerobic exercise. And Clay, my colleague, will go further into that.
Thank you, Jeremy. Not only will the increase of aerobic exercise reduce the amount of inflammation inside of your body, but we also will start to see a difference in the mass of the body and of the major organs. On the table below, we see that the old mice that had no volunteer running have higher masses in almost every single component in body, major organs of the internal body as well, causing them to be at higher risk for things that Jake talked about, like cardiovascular disease. So that brings us to the point of, why do we care? Why is this applicable to our lives today? We all know probably someone that struggles with cardiovascular disease or will struggle with it. Heart disease runs in many families. The ability to find out and hypothesize that aerobic exercise can actually slightly reverse the aging process, lower body mass in overall bodies and major organs, can help us lower those risks of cardiovascular disease. For many of us now at a young age, it's not super applicable until we get older, but the regular cardio or the regular aerobic exercise we do can lead to healthier and happier lives. So I have one question for the rest of you. Would you rather look like this, or would you rather look like that? Please don't be a Hunt, be a Shaw. Thank you.
Just, whoa, just stop. Okay, just